Thank you, Eduardo, and thank you, Herbert, for inviting me to speak in this symposium. We will continue talking about immunity, and the aspect of immunity that interests my lab very much is that it works so well. So we all have gone the experience that we have an infection of, be it upper respiratory tract or gastrointestinal, and within literally 2 days we get better in most cases, I would say 99% of the cases, even if the absence of any therapy. And one of the cells that is responsible for the immune system being so effective is the neutrophil. The neutrophil makes up about 70% of the cells in each one of you. And if there is a disease where there is absence of neutrophils, this is truly catastrophic. There is many infections, and people die of many types of infections, mostly bacterial and fungal. So the neutrophil is there for the first line of defense that we have in immunity, and surprisingly, it's a cell that is very much understudied. So this is a movie I'll show you of a neutrophil that was explanted, was purified from human blood, and in vitro was infected with a bacterium, and here you can see the cell, and you can see the bacteria. This cell is characterized because it has a nucleus that is very polymorphic, and it has all these kinds of granules. And what this neutrophil can do is migrate towards the bacterium, then eat it inside a vacuole, as you will see here. And in all these structures that you see here are granules that contain proteins and proteases, antimicrobial peptides that kill the bacteria that are inside the vacuole. It also produces a lot of radical oxygen species at the surface of the vacuole that contribute to this antimicrobial effect. So this process that you just saw in this movie is called phagocytosis. It was discovered around 120 years ago by Metchnikoff and by Paul Ehrlich in Berlin. And this is probably one of the most effective ways we have of getting rid of bacteria. So we were working with neutrophils, and we discovered that neutrophils have a different way of killing bacteria. So what you see here is a scanning electron micrograph, again, of neutrophils isolated from a healthy donor. And it's kind of a static view of what you saw in the movie. So if you focus, for example, in this cell, this is a cell that is extremely flat because it's actually crawling around the surface trying to find bacteria to eat it. And you can even see pseudopods in this cell that the cell emits in order to catch the bacteria. What we discovered are these extracellular structures that you can see here that the neutrophil makes upon activation, and these structures we think are important in antimicrobial defense, and we call them nets because they come from the neutrophil, that's with the N, and they are extracellular traps. They have an unusual composition in terms of immunity, and what I'm showing here is a confocal image of a neutrophil culture that was stained with different antibodies. So if we take a marker that is typical of the granule, like elastase, you can see that the elastase is localized exclusively to the granules. It's a non-activated cell, like this one, for example, or this one, but it's also present in the nets. And the nets are also stained with DNA and with the core histones, with H2A, H2B, H3, and H4. So the nets are composed of chromatin that has been processed and has been decorated with specific proteins that come from the neutrophil. We've done a proteomic analysis of the nets, and it turns out there are very few proteins in the nets, around 30, and it is a surprise for us, as you'll see in a minute. So neutrophils are cells that emerge from the bone marrow as terminally differentiated cells, that is, they won't divide, and they have a very short half-life, around 6 hours in circulation. So this means that we are unable to do a lot of genetic techniques with the neutrophil, so it's been an interesting experience to work with cells where we cannot use very modern biology, right? We cannot do siRNA screens in a cell that lives 6 hours and won't divide. So I will walk you through a series of experiments where we've tried to understand how nets are made first, and then I'll finish up by trying to show you why we think the nets are important. So the first experiment was, how come a cell can expel its own chromatin? This is unprecedented in biology. And the approach we first took is to do video microscopy. And I'll take 2 slides to explain the experiment I'll show you. We took neutrophils, again, purified from healthy donors. We marked them with one label, which is calcium blue, which is a viability marker. It means the cell is completely blue as long as it's alive, and it will lose the blue color when the cytoplasmic membrane integrity is compromised. This is the first color. The second color is annexin 5, which is a marker for cell death. This is in green. And the trick of the experiment is that annexin 5 is in an extremely low concentration, so undetectable by the microscope, unless it's concentrated on the dead cell, and then the green will appear. So these are the 2 first colors, and there is a third color, and we generated FABs with a monoclonal that recognizes chromatin. We did similar experiment with antibodies that recognize elastase. And these antibodies, FABs, are labeled in red, and they're also in a very low concentration, so undetectable by the microscope until the nets are formed and the antibody recognizes the nets. So I'll show you this movie again. These are two neutrophils that are in blue because they're alive. This is obviously accelerated. This is a process that in vitro takes around 3 hours. And you will see that the neutrophil is first having a process where the nucleus compacts and then expands. And the cell is still alive for another 2 or 3 hours. And then there is a final compaction that you'll see in a second. And then as the cell will lose the color blue and turn yellow and green at the same time. So this was the demonstration. The actually neutrophil has to die in order to liberate its chromatin in a process that which we think is new. So this is again a static view done by transmission electron microscopy of the movie that you saw before. And there is a naive neutrophil that you can easily recognize because it has this lobulated nucleus and a lot of granules. 60 minutes after activation, you can see the shape of the nucleus is very impressive. It's now completely circular. The border between the A and heterochromatin is changed, and if you, in detail, there are many changes in the membrane. For example, the two lamellae of the nuclear membrane are much more separated than before. Now, 2 hours after activation, this is more impressive. This is the remnants of the chromatin, and these vesicles that surround the cell are the remnants of the nuclear membrane. So the nuclear membrane disappears, and there is direct contact of the chromatin with the cytoplasmic components. That's what I said, that we were surprised that the nets only contain 30 proteins, because there is a process by which the proteins that will end up stuck to the chromatin in the formation of nets, and we don't understand this process at all. Finally, 3 hours after activation, the cell is about to make nets. You can see that the modified chromatin is occupying most of the cytoplasm, and we believe this is a cell that the cytoplasmic membrane is still intact, and it will burst in soon after. So how do you study the molecular events of a cell where you cannot do genetics? And this has also been an interesting experiment. And one of the approaches we took is, I'm sorry, before I go there, this is an unusual form of cell death. It's been demonstrated by many investigators that neutrophils can undergo bona fide apoptosis. In this case, we activated the Fas receptor, and you can see that the cell has the classical morphology of apoptosis. There is DNA fragmentation, it's caspase dependent, it's all as it should be. The cells can undergo necrosis, and that is, we induce necrosis by incubating with a pore-forming protein that we isolated from Staphylococcus aureus. And you can see that the morphology is different. The nucleus does change shape, but the nuclear membrane is unaffected. And we believe that the neutrophil can also go through a third form of cell death that we call netosis, which results in this immune activity. So as I was saying, we've been going through a series of different processes to try to understand the molecular mechanisms of how a cell works, how this mechanism works in a cell where we cannot do genetics. So one of the first approaches was on a collaboration with Herbert Waldman that we'll talk later today, and it's been a lot of fun because I've learned a lot. And what we decided to do is to do a chemical screen. We actually believe a lot in experiments, and we did a dress rehearsal with this chemical screen where we take neutrophils, we incubated with 2,000 compounds, then we activated the formation of nets, and then we use an automatic microscope that measures the circularity and the perimeter of the cell to be able to score if the nucleus has changed shape. And these dress rehearsals with only 2,000 compounds actually identify protein inhibitors of the RAS-MEK pathway that was essential for the initiation of the formation of nets. Herbert and I are having a little bit of fun because now we're going for 200,000 compounds, which is an interesting challenge. So this initial chemical screen told us that the first step in the activation of net formation is the activation of RAS, MEK, and ERK. We knew that this pathway activated the production of radical oxygen species in the neutrophil, and the neutrophil is kind of the champion of the production of radical oxygen species. There is an enzyme called FOX that can transport oxygen into superoxide. Superoxide is a molecule that is extremely short-lived and very reactive. It can modify many different, all compounds actually, lipids, DNA, proteins, etc., etc. And then the superoxide dismutates to hydrogen peroxide. Hydrogen peroxide is an antimicrobial. You use it. You can buy it in the the pharmacy, and all of you use it when you have a wound. And this hydrogen peroxide is a permeable member of the radical oxygen species that might be relevant later. And then the neutrophil has another enzyme called myeloperoxidase that converts hydrogen peroxide to these different halic acids. These halic acids are perchloride, for example, that you use to disinfect, disinfect several surfaces, for example. So both hydrogen peroxide and hypochlorite are anti-infectants, so they destroy microbes, although it's still not clear whether they are producing sufficient quantities inside the cell to kill microbes. So let's go back to nets, and what we suspected is that the NADPH oxidase, the FOX, would be relevant, and we did pharmacological experiments to actually demonstrate that, that the FOX is actually important, and it was important. So the problem, another problem of working with neutrophils is that there is no cell line that replicates the activity of FOX, and the mouse is not the great model to do this. So we turned into, to try to demonstrate genetically that FOX was really important. We started working with CGD patients. So CGD stands for chronic granulomatous disease. These are patients that are born with mutations in one of the subunits of the NADPH oxidase, or FOX. These people are, these children are born with this defect, and again, it's a catastrophic disease. They usually die before they turn, they reach the teens, and the reasons are that they are very susceptible to infections. The leading cause